there because they're like a regulation and stuff. So I need
pre-mRNA and regulation of mRNA is here.
Yeah.
Yes.
Yes.
Actually, there are gene regulation. These are two topics.
Okay.
Yes. Yes or no?
Actually, 3. There are regulation of the protein.
And protein activity. Yeah. You know why?
Why?
Here it's very small, but you should explain it a lot. Level of gene regulation in eukaryote. Transcription. Epigenetic regulation. Structure of chromatin. What was the structure of chromatin? Do you remember what do you have in the chromatosome?
Chromatosome? Or nucleus? No.
Yes, in the chromosome we have a nucleosome and
histones.
histone. How many?
They have
8 and 1, 9 histone. Fourth type of histone. Where is this? Chromatosome.
In chromatin, like generally, there are RNA, DNA, histones, ions, like inorganic ions or something.
Yeah. The structure of chromosome. Double DNA string, H1 link them.
Yes.
8 histone here, 8 and 1 is 9 histone. How many type of histone do you know? 4 type, H1, H2, H2, H3 and H4. And H2 has a H2A, H2B. You see H2A, H2B here, H4 and H1.
Yes.
Now back here, you see then in the structure of chromatin, H1 phosphorylation. What's happened if the H1 is phosphorylate? Cannot link the two.
Yeah.
In this case, the chromatin is loose. If the chromatin is loose, not dense, what's happened? The transcription starts very easily.
Is loose here? I thought it was. I thought it was.
What's happened? If H1 phosphorylation lose the dense chromatin.
I think I read this nucleosome when it's phosphorylated. It's
nucleosome also. No, nucleosome or H1 phosphorylated, it means the active. The activation means the loosening of the chromosome structure, and in this case, the transcription enhance. Rule of non-histone protein. What does it mean, non-histone protein?
They're regulatory, like they're like membrane proteins or stuff like.
How many
like
protein
DNA,
do we have?
Polymerase and stuff like that.
We have histone and non-histone. We have yes.
So if you have a DNA and you remove all of the histones, you will be left with the non-histone proteins, right?
Yeah, you are true. But I want to ask you, for example, what is the strong acid? This 6 HClO4 H2SO4. What is the weak acid? Every acid except this. Yeah. What is the non-histone? All of the protein is not histone.
Yeah.
Except histone. Understand?
Yes.
Now, the histone acetylation and methylation. What's happened if the histone acetylate and methylate? Again, the loosening of the dense chromatin transcription enhance.
I think one of if it phosphorylates, I think it became like more dense or something. And if it acetylates, the I don't know, maybe mixing with the nucleosome. The nucleosome, if that acetylates,
you see, remember it like me. Everything added, the density is loose. Everything. Acetyl, methyl, phosphor, okay?
Everything is loose.
If the teacher said no, you can change your sentence.
Yeah.
Opposite. But I'm sure.
Okay.
100% I'm sure, okay?
Right.
Now, DNA methylation in CpG Island. Gene inactivation. If the DNA is methylate, what's happened? Do you know the DNA repair?
DNA repair mechanisms?
How many repair mechanisms?
There are base excision repair.
Base repair, okay.
Nucleotide
excision.
Yeah.
Nucleotide excision, base excision.
Yes.
Again, more. By the help of apnucleones, endonuclease, and something else. It's here. The mechanism of mismatch repair. On the mismatch repair, one of the strands is methylated. Do you see?
Yeah.
Why? Which strand should be methylated? The mother strand. The daughter strand, no methyl. The daughter strand has a mutation, not the mother. Yeah?
Yeah.
And how the recognition protein and endonuclease distinguish between the maternal and daughter? The maternal should be methylated, understand? And it says here DNA methylation in CpG. Island, I don't know what is the CpG, but the DNA methylation, it means gene inactivation. This strand is inactive, just the other one is active and can be endonuclease come and correct it.
So
in the mismatch repair.
the mother strand is methylated.
Mother strand, just mother. Please
Always?
keep this one.
Yeah.
Keep this one.
It's always methylated?
Yes, this is a mother. This is a daughter. The daughter has problem, not the mother, and the mother will be methylated. In this case, the other enzyme can just come here. This is like a barrier. Don't come to me. I'm correct. You should be correct according to me.
Yeah. Like modification methylases.
No, not the modification. It is for the repair, and it's written here.
No, but it's like in prokaryotes, you have this this methyl group that is added, right? For restriction endonucleases, but you know.
Yes, but now we are speaking about just eukaryote, because the eukaryote has a mechanism of DNA repair. The eukaryote has a mechanism of level of gene regulation. Please stop it. Please enhance it. And if the eukaryote lose the control of gene regulation, it means this is a tumor. This is a disease.
Yeah.
Genomic imprinting. What is the genomic imprinting? See, always from DNA you produce RNA. From RNA you produce protein.
Protein, yeah.
If you want to understand what is the DNA, can you use the pattern of the protein?
What is the DNA? Yeah, you can do
DNA,
it. Yeah.
mRNA, and finally protein.
Yeah.
If you know the protein, can you back to the structure of DNA?
Yeah.
No.
You can't?
No. Why? You can back the structure of cDNA. What is the cDNA?
It's,
complementary DNA.
Yeah.
Because when you back in the codon of the protein, you don't have a codon of the intron also. And the DNA has intron. It doesn't have intron. It means from here, if you back, you can produce mRNA. Yes, that's okay.
Yeah, but
here, not cDNA, not the DNA. Complementary DNA. This is the genomic imprinting. This is the genomic imprinting.
From protein to
cDNA. And we can estimate what's happened on the DNA. This is genomic imprinting.
Okay.
Transcription factor also could be enhanced or can be mutated and doesn't do it or do it a lot. It has a role on the gene regulation, on the transcription. Okay, level of transcription is finished. Now we should go to RNA processing. Next topic. Pre-mRNA processing. Alternative splicing. What does it mean, alternative splicing? You learn it.
Yes.
Different shortage.
Oh,
this
yes,
is like that.
yeah.
Okay. It means maybe 2 intron, maybe all of them, maybe one of them. Just omit.
Are removed.
Removed. Omit or removed. Omit.
So you can decide
Omit.
how many introns it can remove.
Yes.
All right.
This is alternative. So one pre-mRNA, maybe several mRNA is produced, and one of them has one job in something, and it has. This is 2 examples of that.
Yeah.
Constitutive splicing. It means one pre-mRNA, just one mRNA,
Yeah.
constitutively. Example for alternative splicing: 1. Calcitonin. Calcitonin is a hormone of thyroid gland. Stimulate deposition of calcium and phosphates ion into the bones. CGRP is calcitonin gene-related peptide. Is produced by certain neuron, not by thyroid gland, by neuron. Cause vasodilation and lowers the blood pressure. Both of them are coded by the same gene. Value are produced by alternative splicing of the same pre-mRNA. See, this is a DNA. Of both of them, CGRP and calcitonin. Exon 1, exon 2, 3, exon 4, 5, 6.
Yeah.
Polyadenylated elements are located at the end of 6 and 4.
Yeah.
Pre-mRNA is here, yes, and maybe my mRNA is finished here at the end of E4, maybe at the end of E6. This is calcitonin, this is CGRP. It produces on the neuron, it produces on the thyroid gland. One mRNA, one pre-mRNA, 2 mRNA produced. One pre-mRNA, several mRNA. This is an alternative splicing by different lengths.
Yes.
But if one pre-mRNA, one mRNA, one mRNA, this is a constitutive splicing, 1.
Yeah, but this one is
Yes,
not.
1 to several is the alternative, and this is an example of alternative.
Okay.
This is 2 of them. This is a calcitonin and this is CGRP. They have the same genetics, same DNA, but different splicing.
This is alternative. This is constitutive.
No, both of them are alternative. We don't have constitutive here.
Sorry.
If you want to produce constitutive, you said exactly same.
Oh, true.
Without difference, just 1. 1 to 1 is constitutive. What here is 1 to 2.
But this one is longer than this one.
Yes, because it has a E6 also. It's finished here. Polyadenylate AAA is here. It's finished here. And just remove E4 and said, I don't want you. I remove all of you.
Okay.
RNA editing, you learn it in, for example, instead of C, put the U and the stop codon happens. Deletion, addition, or substitution of one nucleotide in mRNA.
Yeah.
Again, deletion.
Addition. Or substitution of one nucleotide.
What the deletion and addition? Oh, sorry. Or substitution. If this is 1 or 2 nucleotide, this is the inactivation.
Yes.
But if this is 3,
it's activated.
and if this is 6,
if it's 6, then it's activated, of course.
If this is 4,
it's not.
Understand?
It's one, like, can it be like one of them can be?
No, it means it should be a 3 or something related to 3. For example, 6, 9,
okay,
12, 15. It should be 3 by 3.
Yeah. Okay.
Now, I want to do the gene regulation at the mRNA degradation. mRNA degradation, what is the role of the cap? Protection from degradation.
Yeah.
We need poly-A tail, 3' non-translated region. We need binding protein also. Every of them has a problem. Maybe degradation doesn't happen, maybe happens a lot. It depends, okay, the situation. Translation. Now we produce mRNA. Maybe mRNA degraded if it doesn't have a cap. Now we should coming here out, transport it out. For the transport, we need the nucleocytoplasmic RNA. Export can be regulated. Nucleocytoplasmic from nucleus to cytoplasm. The export could be regulated. For example, maybe the channel here is closed and said, where do you want to go? Please back. So this is the transport regulation. Degradation, you learn it. This is the mRNA degradation. If not, translation happens. If the mRNA doesn't degrade, the protein translation happens. Okay? Now, and the level of translation, phosphorylation of initiation factor. For example, microRNA. What's happened? If the initiation factor, where was the initiation factor? They're coming to the 30S subunit by GTP. Do you remember?
Yes.
If they phosphorylate, they cannot do the job properly. In this case, translation is stop. Now, in the case of stop translation of bacteria, that's good. But at the level of the eukaryotic cell, it's not good. If this is not a phosphorylation happens and never phosphorylation happens, what should we do? This is a tumor because a lot of protein is produced. For example, somebody has a thyroid gland and a lot of thyroid is produced. Why? Because of the phosphorylation, non-phosphorylation of initiation factor never happens.
Okay.
Because when we need, we shouldn't, if we need, we should dephosphorylate. When we doesn't need the extra hormone, we should phosphorylate and say, please stop the job. If we cannot stop them, they produce more and more and more protein.
So you don't need it, you phosphorylate it.
Yeah. Mechanism of action similar to siRNA, small interfering RNA. What is the job of small interfering RNA?
It regulates the gene expression.
Could you please take this? See, this is, I am a siRNA, okay? I come in, I'm exactly like that. And this is the mRNA. I come here and I degrade it. It's nothing that you see. Where is it? It's chapter 9.
Yeah. You have endogenous and exogenous siRNA that can be.
Yes, I think so. siRNA, yes. You see, siRNA exogenously come, make a silencing complex. Maybe endogenous coming and they come.
But
and
they work
kill
with the protein glyco or something. Protein.
Not important. Just important that siRNA, the job is degradation.
Yes. Okay.
This is a one level of what?
Protein degradation.
One level of regulation. One level of regulation.
This is the first regulation factor, or.
You see, now I'm here. I want to regulate between mRNA to protein. Okay. I said I send siRNA to degrade you, and you don't produce any more protein.
But where does the regulation starts?
From here, from DNA transcription.
From here.
You see, this is central dogma. At every level, we have a first at the transcription, then we go to RNA processing, maturation, then RNA transport, mRNA degradation, and now we are here on on the branch of translation.
But how can it be regulated by pre-mRNA processing? Like, how can it be regulated?
How could be regulated?
Yeah.
Doesn't happen splicing here.
Okay.
Or maybe happens a lot.
Okay. So the regulation, like here, it starts from here?
I don't understand what you say.
You know, it says like regulation of pre-mRNA synthesis, right? So
mRNA synthesis or processing?
I think it's
Processing.
processing.
Synthesis and processing.
Synthesis and processing.
Yes, this is a topic number 48.
Mm-hmm.
Topic 48. And then it's a regulation of mRNA transport, translation, and degradation.
Mm-hmm.
It's from here, topic 49. Okay? And then is a protein activity and degradation. Topic 50. But you should explain a lot. What is the translation in eukaryotic, in prokaryotic, okay?
Okay, but I'm wondering, like, you know, you said about siRNA?
Mm-hmm.
That's a regulation? Like, it can be regulated by it?
You see the siRNA where we are now?
It was...
Here, siRNA.
Yeah.
The mechanism of action of microRNA is like siRNA, because siRNA degrade mRNA, degrade mRNA, and here microRNA also degrade mRNA. But this mRNA at the level of translation, that mRNA is free. But actually they are same. Both of them, micro and SI, small interfering, degrade the mRNA here. No, protein degradation, maybe the protein is produced. No, I should protein maybe degraded or maybe activate. Protein degradation by lysosomal proteolysis, autophagosome, and phagolysosome. What is the autophagosome? When the lysosome eat itself.
Its own organelles?
Or our organelle.
Like autolysis?
Yes. And phagolysosome, it means, for example, see this is a protein. Protein is produced abnormally. The lysosome come, phagocyte that, and phagolysosome produced. Ubiquitin proteasome pathway. Protein with destruction box, polyubiquination, degradation by proteasome in cyclin B in anaphase. Just know this is a destruction box. Do you know the cell cycle?
Yeah.
What is the cyclin B? Is in APF or MPF?
It's in it's in MPF.
MPF.
I just needed a second.
Sorry. Do you remember this?
Yeah. What is this?
What is this? I ask you.
Oh,
it's mitose.
Yeah.
Why the MPF is degrade here? Because of destruction box. Polyubiquination happens here.
But what was a ubiquitin again? What was that again?
Just ubiquitin.
Yeah.
It's a proteasome pathway. It causes the killing and degradation of protein.
Okay.
If anywhere happens, ubiquitin. Okay? And this is a drug. We inject polyubiquitin to kill the product of the tumor cell, to don't metastase any other way.
Oh, okay.
Monoubiquitin is a polyubiquitin. Now, monoubiquitin, no protein degradation, has regulatory function. Again, no protein
degradation
has regulatory function. It doesn't cause, just regulatory.
But the poly, it does?
Yes. Degradation by proteasome, for example, cyclin B in anaphase of mitosis happens. What is the protein function? Allosteric regulation by a small molecule. Allosteric. What does it mean, allosteric? See, for example, I am a protein here. I am a G protein, for example. This is a GTP. GTP comes to me, plus me, what's happened? I modify the shape of me. It means I am allosteric. Okay. Allosteric means modify the shape. Other means of the allosteric, we have an inhibitory allosteric and induction allosteric, inducer allosteric. It means I don't have a special receptor for you. But I said, do you want to come to me and encourage me? Okay, welcome.
Okay.
I don't have any inhibiting site for you. You cannot inhibit me. But I said, okay, if you inhibit me, are you satisfied? You said yes. Okay, come and inhibit me. Understand? Allosteric protein doesn't have a specific receptor for anything. Just anybody come, induce them, anybody come, inhibit them.
Okay.
Allosteric. Covalent modification happens by the protein. Protein could do the phosphorylation, for example, protein kinase and phosphatase. What does it mean, protein kinase? Kinase or kinase means add phosphate.
Yeah.
Add
Phosphate.
everywhere, Aisha. 99% phosphorylation means the activation. 99%. Okay, it means phosphorylation. But kinase also means phosphorylation. This is synonym. But this is activation. This is a 1%. Now, how many type of kinase do we have? You learn it later. Serine, threonine, and tyrosine, and serine kinase. Protein-protein interaction. Maybe cyclin by CDK complex may do the interaction. For example, cyclin B by CDK, it make a MPF, and cyclin E or D make a APF. One of us, other one. Do you remember? No. Come back here. You just said means I don't know.
No, I remember. I remember the SPF.
Oh, SPF.
And MPF. I know these two. Because the B
no,
and...
you should know exactly the name of the cyclin. On the SPF,
yeah, see, this is B
cyclin B and CDK1.
Yeah.
This is the MPF. And on the SPF, we have a cyclin E and CDK2.
This one.
You should know this number and name of the cyclin. Cyclin A2 or E2 is the SPF. Cyclin B1, cyclin B and CDK1, B1 is the MPF. MPF is B1, SPF is E2. Do you get it?
E2, B1.
B1. This number is important. If you don't know, you will fail.
I will. I know it. I just have to draw it first.
Okay.
Because I have a system.
And I really think that